Hey everyone. Welcome to my new channel. Today, I'm going to share something that's genuinely changed how I approach reading research papers. And trust me, if you're in academia, doing research, or preparing for journal clubs, this is going to save you hours. We are talking about an AI prompt that helps you systematically break down any research article. And I mean, any field. Whether you're in biology, chemistry, engineering, or social sciences. This works. Now, I know what you're thinking. Another AI trick? But hear me out. This isn't about letting AI do the work for you. This is about using AI as a structured reading assistant, one that helps you extract the most important information quickly and systematically. So, here is the plan for today. First, I'll walk you through the prompt structure and explain why each section matters. Then, we'll do a live demonstration using a real research paper. Specifically, a recent open access Nature article. And finally, I'll show you exactly how this helps with journal club preparation. Let's dive in. Alright, so here is the prompt. Now, I'm going to walk through each section, and I want you to think about how this maps onto your typical reading process. First, notice how we are setting this up. To make an AI prompt effective, there are 6 elements that people often overlook. These are role, task, context, constraint, format, and example. First, we are giving the AI a role, acting as an expert researcher in your field. This is important because it primes the AI to think in that domain-specific way. Then, we have the task. Analyze a paper according to a framework. The key word here is strictly. We are not asking for creative interpretation here. We want structured analysis. The context section, and this is crucial, is where you input your specific research focus. Maybe you're working on AI applications in cancer drug discovery, or sustainable materials for energy storage, or social media's impact on mental health. Whatever it is, you put it here. This makes section 4 of the analysis specifically relevant to your work. And the constraint. If information is missing, state not mentioned. Do not hallucinate data. This is so important. We are telling the AI, be honest about what is not there. Don't make stuff up. Now, let's talk about the framework itself. The framework provides the format and example for the AI to follow. It's divided into 6 main sections, and each one serves a specific purpose. Section 1. Bibliographic and metadata. This is your quick reference. You get the proper citation, a 1-sentence TLDR, just 30 words to tell you what this paper is about, and keywords. Think of this as your paper's ID card. Section 2. Research foundation. Here is where we get into the why. What problem are these researchers trying to solve? What is their hypothesis? This section answers. Why does this paper exist? For journal club prep, this is important because you can quickly articulate the motivation to your audience. Section 3. Methodology and design. This is the how. What did they actually do? What tools, techniques, or models did they use? Notice we are asking for specifics. Not just they used mice, but how many? What selection criteria? What platforms? This level of detail is exactly what you need for journal club when someone asks. But how did they control for X? Section 4. Key results. This is the meat of the paper. And look at how we've structured this. Core findings. Key data points. And we are emphasizing, use numbers. No vague statements. A table summary of figures, which is brilliant for journal club prep. And auxiliary results. This section is designed to give you the factual backbone of the paper. Section 5. Critical analysis. The so what? This is where it gets interesting. We aren't just summarizing, we are critically engaging. What do the authors conclude? How does this relate to my research? Remember that context we set earlier? What is the actual contribution to the field? What are the limitations? And what opportunities does this open up? This section basically writes your journal club discussion points for you. And section 6. References. Finally, we ask for the 2 most important citations you should read next. This creates a pathway for deeper understanding. So, that is the framework. Now, let's see it in action. Alright, I'm going to use Google's Gemini 3 Pro for this demonstration. And of course, you can use ChatGPT, Claude, or other models. But I found Gemini handles long papers particularly well. The paper we are using today is a recent Nature publication. And I want to emphasize, this is an open access article, which means anyone can read it. The title is Transient Hepatic Reconstitution of Trophic Factors Enhances Aged Immunity, by Mirko Friedrich and colleagues. Published in Nature around 2 weeks ago. And it is a really fascinating study about using the liver to improve immune function in aging. Now, I am not an expert in immunology. But that is actually the point. This prompt helps me understand papers outside my core expertise quickly. I'm now attaching the PDF, pasting the prompt, and for the role and context section, let me put something like, act as an expert academic researcher in the field of immunology. And my current research focus is, for example, the impact of aging on immune systems. Same goes to my research topic and the contribution. And let's see what we get. Perfect. Look at this. Let me walk you through what we've got. Section 1. Bibliographic and metadata. So, we've got our full citation here, ready to copy and paste into your reference manager, except it is lacking the issue number and DOI code. The TLDR says, by repurposing the liver to transiently secrete thymic factors, specifically DLL1, FLT3L, and IL-7, via mRNA LNPs, researchers successfully rejuvenated the T-cell repertoire and boosted anti-tumor immunity in aged mice. In less than 30 words. I now understand the core premise. For Journal Club, this is your opening slide. And keywords include immunosenescence, thymic involution, mRNA therapeutics, synthetic biology, T-cell rejuvenation. These immediately tell you what domains this paper touches. Section 2. Research Foundation. The core problem. Aging leads to thymic involution, or shrinkage, reducing the output of naive T cells and impairing immune responses to infections, vaccines, and cancer. Previous attempts failed because of toxicity and lack of feasibility. See how specific that is? Not just aging is bad, but precisely what about aging they are addressing. Their hypothesis? The authors hypothesized that the liver could be repurposed as an ectopic biofactory to produce essential thymic trophic factors. By restoring them with mRNA-loaded LNP, T cells can be rejuvenated without significant toxicity. For your journal club, you now have your introduction and hypothesis slides basically written. Section 3. Methodology. Look at this breakdown. Study design, we have discovery via multiomic profiling, engineering of mRNA constructs, and what validations were done in aged mice, specific and straight to the points. For data sample, they profiled 97,000 circulating T-cell transcriptomes, 47 spatial arrays, and scRNA-seq of tumor-infiltrating lymphocytes. Key tech includes types of transcriptomics used, delivery system, computational tools, synthetic biology. This is exactly the level of detail you need. When someone in Journal Club asks, how did they deliver these factors? Boom. You have it. SM-102 LNPs. Section 4. Key results. Okay, this section is comprehensive. Let me pull out some highlights. Core findings. The DFI mRNA cocktail successfully reprogrammed the liver to secrete functional immune factors. This expanded common lymphoid progenitors and boosted naive T-cell numbers in aged mice, reversing myeloid bias without affecting HSC composition. Key data points includes vaccine response. DFI-treated aged mice showed an approximate two-fold increase in antigen-specific CD8 positive T cells. Tumor survival. In the aggressive B16 OVA melanoma model, DFI plus anti-PD-L1 checkpoint blockade resulted in 40% complete rejection in aged mice. See those numbers? 40% complete rejection. That is what we want. Quantitative statements. And look at this, the AI has summarized the key figures. For Journal Club, you can literally go figure by figure using this table. Figure 1. Identified decline in Notch, FLT3L, and IL-7 pathways. Confirmed liver expression of mRNA-delivered factors. That is your Figure 1 slide summary right there. Section 5. Critical analysis. The so what? Alright, this is where it gets really valuable. The conclusion. The liver can be temporarily engineered to act as an auxiliary thymus, providing the necessary trophic factors to rejuvenate the aged immune system, improving responses to vaccines and cancer therapy with high safety profile. This is the punchline for the article. Relevance to my research topic. Look at that. This paper is a landmark study for your interest area. It moves beyond describing immune aging to engineering a reversal. It directly addresses the bottleneck of thymic involution. It isn't just summarizing, it is analyzing relevance to my stated interest. For the contribution, it has summarized it in 3 aspects: methodological contribution on proving ectopic expression as a strategy to bypass thymic atrophy, technical contribution on complex cocktail delivery with mRNA LNPS, as well as therapeutic contribution on cancer immunotherapy in the elderly. This provides a broad overview on the whole paper. Limitations. Transient effect. The benefits fade after cessation of dosing, effects reversed approximately 4 weeks post-treatment. This implies chronic or repeated dosing would be required. Or, human liver physiology and immune aging are different from mice. These are your discussion points. These are the questions you will raise in Journal Club. Future opportunities. Could we engineer a smart LNP that only expresses these factors when it detects markers of severe immune suppression? Or, as well as the possibility of combination therapy and application of AI? This is brainstorming material. This is where new research ideas come from. Section 6. References. And finally, we get 2 key papers to read next. Middlebrun and Cromer, on hallmarks of T-cell aging, and other related literature by Ross et al. On depleting myeloid-biased hematopoietic stem cells to rejuvenate aged immunity. This creates your reading pathway. If you want to go deeper, start here. So, let's talk about why this prompt is so effective. First, structure. It forces systematic reading. You can't just skim and hope you got it. Each section demands specific information. Second, quantitative focus. Notice how we kept emphasizing. Use numbers, don't be vague. This prevents that thing where you read a paper and come away thinking, they found that X was better, but you can't remember if it was 10% better, or 10-fold better. Huge difference. Third, critical engagement. Section 5 isn't just summary, it is analysis. It asks, so what? It asks about limitations. It connects to your work. This is what separates a good Journal Club presentation from just reading the abstract. Fourth, scalability. Remember, this works for any field. You just change the context section. I used an immunology paper, but this exact same prompt works for computational linguistics, material science, whatever. For Journal Club specifically, look at how this maps out. Section 1. Your title slide. Section 2. Your introduction and hypothesis slides. Section 3. Your method slides, with the detail needed to answer questions. Section 4. Your results slides, using that figure table as your guide. Section 5. Your discussion and critical analysis. And Section 6. Your future directions or related work slide. You basically have your entire presentation outline. And here is the thing. This doesn't replace reading the paper. You still need to read it. But this gives you a framework, highlights what to focus on, and ensures you don't miss critical details. Alright, so that is the prompt. I really hope this helps you in your research and Journal Club prep. A few final thoughts. 1. Customize the context section. Make it specific to your research. The more specific you are, the more relevant Section 5 becomes. 2. Use this as a reading guide, not a replacement. Read the paper yourself, but let this prompt structure your reading and note-taking. 3. This prompt is available in the description below. Copy it, modify it, make it your own. Now, before we wrap up, if you're interested in staying updated on the latest breakthroughs in biology and biomedical research, I'm actually doing something new. I'm creating podcast-style deep dives into recent discoveries on both Substack and Patreon. We go beyond just the headlines, we talk about the implications, the methodology, what it means for the field. If that sounds interesting, links are in the description below. I'd love to have you join that community. And if this video was helpful, do me a favor. Hit that like button, subscribe if you haven't already, and drop a comment telling me what field you work in. I'm genuinely curious what research areas are represented in this community. Alright, that's it for today. Thanks for watching, and I'll see you in the next one.